Hello AP biologists, welcome to Enzyme Lab Prep. Usually I spend this time by myself, but this year I'm inviting you into it so that you can see exactly what into prepping your enzyme lab. Let's start with this jar right here. This is catechol, our substrate for the enzyme that we're going to be looking at. And catechol is probably the most toxic chemical we'll use all year. I'm looking at a label that says, warning, this product contains a chemical known to cause cancer, birth defects, or other reproductive harm. So while we're using catechol, we need to make sure that we're wearing gloves and goggles. I made this catechol into a solution for us so that it's actually dissolved here in this water, and then we'll put it into solution with our enzyme from our friend, the potato. So the potato, inside this potato are millions of cells that all contain the enzyme polyphenol oxidase. This enzyme is present in that giant central vacuole of the cell, and when I cut into the potato, and then eventually when I blend the potato, it will break those cells and the central vacuoles inside them open. When I break that open, the polyphenol oxidase will come out, and it will be in the solution that can then react with the catechol. So, now you get to watch me prep the potato. This is the most scientific part of the video. You'll get to see me using a potato, a knife, and a blender to make the solution that you'll use in class. Let's go. All right, I sped this part up so you don't have to see me cutting potatoes for a million years. But I just want to reiterate, the reason why we're cutting the potato down is so that we can make a potato solution that has enzymes floating in it freely. So we'll open up those cells, have those enzymes, and we won't have to deal with the whole potato, rather just a solution of potato extract. So I'm actually going to cover the solution with water so that it turns really into a solution rather than potato mush. All right, now we have our solution of water and potato. We're going to put it on our blender, a helpful scientific tool for us today, and blend it. All right, what I have now is basically a potato smoothie. It's blended potatoes and water. So all those potato cells have been broken open, and our enzyme, polyphenol oxidase, is floating around inside here. So in order to isolate just the solution that has the enzyme that we want, I'm actually going to filter this to get all of the excess stuff out. So I've got my filter here with some filter paper, my beaker. We're just going to pour this in to get out the solution that we want. All right, the next thing that I'm going to do is show you the computer setup that we'll use to record our data so that we can see the progress of the reaction. So I've got my parts here that are out in front of us, and you will get three things. You'll get a cord, you'll get this box called a LabQuest Mini, and you'll also get this thing which is called a colorimeter. And what the colorimeter does is it measures how much light is absorbed by the substances in these things that we call cuvettes. So this is a cuvette. It's a small container which contains liquid, and when we put it inside the colorimeter measures how much light is absorbed by the substances inside here. So this is how you set this up. You take your LabQuest and the cord, and there's a small USB end just like the one that's in your phone. You stick that over here on this end, and you plug the other end into the USB port on the computer. Then you take your colorimeter, which has this weird connector right here, and you plug it into one of the channels on the front. They say channel 1, 2, and 3. So I'm going to plug them in. And then I will open this program called Logger Pro, and I'll show you this on the computer in a little bit more detail. And the computer will automatically recognize that you have hooked up your colorimeter. The next thing that you need to do is for our colorimeter, it can absorb light at different wavelengths. And we're going to be observing at the 430 wavelength, which is this last one over here. And so there are some buttons on the colorimeter that I push to get it over to the one that I want. So I'm going to push the button over once. And now you can see that the light moved. So we're now measuring at 430 nanometers instead of 470. For everything that I do, I'm going to be using a cuvette, and I'm also going to be using lids on the cuvettes. And we use the lids in order to make sure that no liquid gets inside the colorimeter, and it stays clean and safe for next year's use. The first thing that we have to do when we use the colorimeter is what we call a blank. And a blank is what tells the colorimeter what to pay attention to and what not to pay attention to. So you can see, actually maybe you can't see, but from my eyes right here, this catechol solution has a slight color to it. And we don't want the colorimeter to measure the color of the catechol solution. We only want it to measure the changes that come about from the reaction with the enzyme. So what we're going to do is actually fill our cuvette with this and do what's called a calibration. The calibration will tell the colorimeter, this is the natural color of the catechol solution. Don't count that, just count everything else. This is kind of like doing a zero on a balance, right? Where if you have to put down a piece of paper or a container that you're going to weigh something on, you tell the balance 0. This weight isn't important, only what I add to it after is. Same concept. So every time that we use the cuvettes today, we're going to make sure that we put 3 milliliters of liquid inside the cuvette. I have pipettes right here, and these pipettes are 1 milliliter pipettes. There's a line at the top. So in order to do that, I squeeze all the air out of the bulb at the top. I fill it up to the line for 1 milliliter, and I drop it here into the cuvette. So I'm going to do that 3 times. If I don't have 3 milliliters of solution, then that won't be enough for it to do the reading. So I'm going to put 3 milliliters of 3 milliliters of catechol in there. And then once I've done that, I'm going to put the cap on. Put the cap on. Next, I'm going to dry off this cuvette just because it looks like a little moisture got onto the outside. I want it to be nice and dry. When you see your cuvette, you'll see that there's a side that has ridges and a side that is clear. We want to make sure that we put the side that is clear toward this arrow at the top of the colorimeter. So the clear side I'm putting toward the top of the colorimeter, and then I close the top. Once I've done that, in order to zero or calibrate my colorimeter, I press the button in the center which says Cal. I press it, the red light will blink at me. The first thing that we'll do is the control. The control is what normally happens in this situation, and we'll be able to compare everything else to the control. So our control today is just what happens when we put catechol and enzyme together in a cuvette. So once again, I'm going to have 3 milliliters of catechol. So 1, 2, and 3. And then I'm going to add 5 drops of enzyme. So I'm going to put this over 1, 2, 3, 4, 5. 5 drops. Next thing I do, as soon as I do that, I put the cap on, and then I invert it. Invert meaning going upside down, right side up. I don't shake it, I just invert it to mix those things together. Then as fast as I can, I put it in the colorimeter facing the arrow, close the top, and then I hit the button that says collect on my computer screen. Immediately, it will start taking data for me. So the computer program will take the data for me on its own. I'm going to let this run for 5 minutes, and then we'll see the results. So I hit collect on the screen, and immediately the computer starts to take the data. It's programmed to take a data point every 3 seconds, so it will measure the absorbance every 3 seconds, and it will simultaneously graph it, as you can see. So I've got time on my x-axis and absorbance on the y-axis. As it says in your lab packet, the absorbance is a measure of how much light is absorbed by the liquid that is inside the colorimeter. The darker the color, the more light gets absorbed. So as the reaction goes on and more quinone is produced, the solution inside the cuvette becomes increasingly brown. The quinone has that brown color, so it's going to become more and more brown, which means that more and more light will be absorbed. So we can see that the absorbance is steadily increasing over time, and that's a direct result of the reaction that is happening inside the test tube. The computer will automatically shut off at the end of 5 minutes, so we can just let this run until it stops, and then in a few moments we'll see that the clip actually stops automatically. And ready, and stop. There it goes. To see the data more clearly, we can rescale this graph. So what I'm doing right now is right-clicking on the y-axis where it says absorbance and hitting auto scale. And that will change the graph so that it's just a little bit clearer for us to see. All right, So I've done my first trial, and you can see that I've got a brown color here in the cuvette, and that's because all of our catechol was converted by our polyphenol oxidase enzyme from the potato to quinone, which has a brown color. So now I'm going to do my next treatment. We're going to be doing a series of treatments, which are just experimental conditions that are different from the control, and we'll compare them to the control using our data. So the treatments each require the cuvette to have 3 milliliters of liquid in them, no matter what we change. So one of our treatments is to use a reduced amount of catechol substrate. So I'm only going to use half a milliliter instead of a full milliliter. So I'm going to fill it up to the half line on my pipette, put that in, and then I'm just going to put distilled water in for the other 2.5 milliliters so that I still have a total of 5. I'm going to do 1, 2, and an additional half. Then I put in my 5 drops of enzyme. 1, 2, 3, 4, 5. Immediately I put the cap on. I invert it upside down, right side up. I find my clear side, put it toward the arrow here in my colorimeter, and hit collect on the computer. Once we do that, it will start taking data, and I will let this treatment run for 5 minutes as well. All right, ladies and gentlemen, when you are done with all of your treatments, take your cuvettes and empty them into a waste beaker. There will be waste beakers around the room. We actually can't dump this stuff down the sink because it contains that toxic catechol. So please do not empty any of your cuvettes down the sink. Empty them into an empty beaker. Once I've done that, then I can go and rinse them in the sink because there will be so little catechol left that it will be okay. But please, please, please do not just dump these down the sink. Once you've cleaned up all of that stuff, your cuvettes are nice and clean for the next experiment we do with this. Please disassemble all of your technological equipment, put it back in the boxes, and then we'll be done with our lab. Make sure you complete all of the pre-lab in order to be able to participate. Email or text me if you have any questions. See you later. Bye.